Umar Ahmed is a senior scientist at the Africa Center for Disease Control. I've known him for a very long time, so it'll be interesting to hear what all the science has been doing, and this will be of interest to the African biogenome community. Dr. Ahmed, over to you, please.
Okay, thank you so much, Dr. Tangat. Can you hear me?
Yes, please carry on.
Okay, let me share my screen. Sorry. Can you confirm you are seeing my screen, please?
Yes, please. You can see your screen. Do you want to make it a slideshow, please?
Yeah, sure. Are you seeing my screen now?
Yeah, please go ahead.
Okay, thank you so much for having me this morning. My name is Umar Ahmed. I'm the Senior Technical Officer for Pathogen Genomics, coordinating genomic sequencing and bioinformatics across West Africa, and at the Center for Laboratory Diagnostics and Systems at Africa CDC. Today, I will be walking you through how genomics can be applied, to strengthen biodiversity surveillance and public health resilience in Africa. So biodiversity, you see, we have three different looping between biodiversity loss, emerging diseases, and climate change. These three components are actually what we call anthropogenic drivers that are caused actually by fossil fueling use, deforestations, and increased contact with wildlife, usually the cause of destabilizing the ecosystems that we have currently. And because of these interactive activities, these three looping that is happening, as you see in the cycle, a kind of cyclical looping between the three: biodiversity, climate change, and infectious diseases, is actually the cause of most of the emerging and re-emerging diseases that is happening. So this disturbing creates really a conditions for pathogen spillover, and also novel diseases emerging and also threatening both diversity and human health. So I will start by telling you the One Health approach for the West African countries, since Nigeria is part of the West Africa. Generally, we have a lot of zoonotic diseases outbreaks in Africa, but particularly concentrated in the western part of Africa, which are actually the convergence or the cause of the humans, animals, and environmental health associations that causes all these pandemics and epidemics, diseases to be coming up. Particularly if you look at the West African regions, we do have kind of resurgence, emergence, infectious diseases like Lassa fever, Ebola, anthrax, Mpox. They are frequently been coming out by the day in the country, particularly in West Africa. So, and all this as a result of human-animal interactions, as well as the environment, that targets or that affected the agriculture, markets, and also the settlements where we are. So genomics can actually be used, to integrate these tools and also connect the surveillance across all the sectors, all the three sectors we mentioned. That is the One Health, humans, animals, and environment. So what is the roles of genomics in linking this biodiversity across all this species? So genomics actually can be used, for tracking pathogens, evolutions, emergence and re-emergence infectious diseases, and also identifying some reservoirs, vectors, and also transmission routes or patterns of a disease, and also spill over in the early onset or early warning signing from the environment when you carry out a kind of like a metagenomic surveillance. That can also reveal how biodiversity alter pathogens dynamics, and also can support the One Health approach by integrating data, species, and ecosystems. So you can see the diagrams from the left side. You get to understand how these biodiversity landscapes, the looping systems that I showed previously in the previous slide, are connecting together, to cause the emerging and re-emergence of the diseases within the biodiversity ecosystems. So at Africa CDC, what we do, what is the genomic status at Africa CDC, and how does it impact, or what is the status across the African countries, actually lies based on the strategic plan that we have from 2023 to 2027. Africa CDC actually ambitions to develop a surveillance systems and an early warning signing systems through genomics applications to support the early detections, characterizing the pathogens, and as well as reporting the outbreaks as early as possible, and also apply clinical services on infectious disease and non-communicable diseases. The aim is actually to strengthen the clinical and public health laboratory systems and network within the continent. Particularly, Africa CDC developed what we call foundational genomics capacity within the African countries. If you see the first objectives, we started this in 2022 to 2023. We developed this capacity within the Africa CDC lab networks. And from there, we, encourage member states to have access to diagnostics, for effective detections, management, and control of pandemics. And also, we also do a lab mapping to identify the capacity of the labs, and also, what are their strengths in terms of surveillance, early detections, and also response to public health. For genomics, particularly, we actually developed the capacity of the member states, through molecular genomics and bioinformatics. We enhance their capacity by providing some genomic sequencers, reagent support, establishing linkages, sample referral systems, and all that. So when Africa CDC started, that was in 2017, there was nothing like NGS capacity in Africa based on the foundational genomics assessment that we carry out at Africa CDC. That was in 2018, 2019, when Africa CDC decided to establish the pathogen genomics unit. From there, we identified that in the entire Africa, we only had 7 countries or 7 member states that have functional genomics capacity, which means they can be able to sequence specific pathogens at a particular level, but they don't have, capacity to do the analysis. And what we identify, we are actually limited sequencing and data infrastructure, lack of skills workforce in the labs, and also the bioinformaticians are lacking in the continent, lack of policy and frameworks, no coordination of sequencing activities completely, challenges with respect to supply chain cost, and also regulatory challenges, when you send some reagent to some countries. These are the, the pre-assessment that was administered by the Africa CDC. That was in 2019, 2018, 2019. With support from some partners, we were able to come up with, the problems. And those problems help us to establish through APRO network, we establish Africa CDC lab networks and regional sequencing hub. As you can see in my screen, we cluster the specialized genomics center. The western part, we have SGIT, which are based in Nigeria, Redeemers University. We have Sanbi in South Africa, ERI in Kenya, and CRISP in South Africa as well. So we cluster those countries with little or no capacity to some specific institutions that we call regional sequencing hub. For example, like in Morocco, we cluster Libya, Egypt, in the North Africa, to join Institute FASTA Morocco, because they have, sequencing capacity. In West Africa, for example, in Dakar, we, linked most of this laboratory in Gambia, Mali, Burkina Faso, Guinea. We link them with Senegal, to support them. If they have a little or no capacity, let's say they have pathogens in their country, and they are unable to do the sequences, they repair the sequence to, IPD for sequencing, and IPD could support them with analysis, even capacity building and support. All the rest of the countries, this is how we establish the networks and we leverage on the APRO laboratory networks and regional sequencing hubs. But we are particularly focusing on the national public health institutions and regional sequencing hub across the continent. And in 2021, the Africa PGI, the pathogen genomics accelerates really the sequencing of the SARS-CoV-2 genomic sequencing in Africa. And that really accelerates three major key pillars that we have on specialized centers and regional hubs, as you see. And also we democratize and expand the sequencing national reference labs, as well as the national public health institutions. We network across the continent with regional hubs and specialized centers. So what PGI basically does, the functions of PGI, is to support the cross-border sample referral system. Initially, it started with SARS-CoV-2, and now we increase it to beyond SARS-CoV-2, including the use cases. We supported at that time 35 member states, with capacity to be able to monitor the variant of public health, the variant of the COVID for public health actions. At that time, we supported the shipment of 34,000, SARS-CoV-2 specimens in the continent. And we supported genomics and bioinformatics workforce. At that time, we conducted over 21 trainings. Over 600 trainees were trained across 52 African Union countries, and we focus more on the use cases that are frequently bedeviling in the continent, like the COVID-19, Ebola, monkeypox, and cholera. We also supported the countries with supply of equipment and reagents. We supported 32 countries with sequencers, 182 shipments of reagents to different countries, and also 200 kV of reagents, which 50% are actually procured, while 50% are donation from partners. And also we supported the quality assurance, the QMS, from the national public health institutions. So we had about 86% passing rates of the QMS for genomics quality assessment before the data can be uploaded to, sequence repositories. So these are basically the contributions of the Africa CDC and the African Union. We contributed to 53 out of 55 member states with sequencers. And the sequencing equipment were actually 70% procured by the Africa CDC, 30% were donations. And we focus on 5 use cases: COVID, Ebola, Mpox, and cholera. And as well as we spend quite a number of money in, procuring some reagents across the continent. So what was really the gaps that was identified post-pandemic were limited molecular capacity, inadequate infrastructure and supply in most of the African national public health institutions, shortage of competent bioinformaticians and bioinformatics scientists who can analyze the data that was sequenced in those regional sequencing hubs. Wide varieties of pathogens, because there are dozens of public health events every day. You have, pandemics here, pandemics there within the country, so within the continent. And non-resilient and non-adaptive, unintegrated health systems that we have in the continent, and silo and fragmented surveillance effort, we are all the key challenges that we recently identified. So that was what brought to Africa PGI 2.0. Before, where Africa CDC pathogen genomics initiatives, we are having 5 pillars that focus more on capacity buildings, sample referral systems, quality management systems, bioinformatics and genomics, training for all the public health and national public health institutions. But after identifying the gaps that we had, we sat down and come up with what we call the PGI 2.0, which specifically focus on 3 components: the surveillance, the laboratory components, and bioinformatics and data analytics, which are what we term integrated genomic surveillance for the public health actions. Without a very effective surveillance systems, there could not be effective laboratory systems that can carry out sequencing, and without sequencing, we cannot be able to have a quality genomics data that can inform public health decision. So this PGI 4.2.0 was actually focusing on 4 different priorities. The one is actually to expand on the foundational capacity. Since in 2018, 2019, we assessed the member states, we were able to understand their limitations, and we build, we build their capacity gradually, and then we were able to, start sequencing some specific pathogens. So we are now moving into expanding those, foundational capacity that we built across the member states. And secondly, we are actually implementing the integrated genomic surveillance systems for early warning sign-in systems that involves making use of the wastewater surveillance system. We sequence even the wastewater to identify if there are, emerging pathogens or the pathogens that have the potentials for pandemic. We inform public policy on it. And then priority 3, we promote timely sharing of quality assured data through what we call pathogen data sharing platforms that is currently being developed by Africa CDC, where it is called AGARI Platform for Public Health Actions, where we ask member states to submit some data onto the platforms and, ensure we promote the timely sharing of the quality data for genomics. And that will help country, to like a wake-up call for early warning signs and detections of pathogens with pandemics. And we also create and enable these systems with sustainability mechanisms, frequently being carrying out advocacy, policy, governance, and also funding sustainability structure. So the 2.3, 2.0, the integrated genomic surveillance are actually based on response, samplings, analysis, and also the bioinformatics as well as the testing. So basically, if you look at here, is when we have the samples, we normally usually ask the member state if they have the capacity, they can sequence the samples, even perform some molecular diagnostic testing at the labs, and then from there, if they can prove they can test the pathogens, then it's fine. If they cannot, we refer them to regional sequencing hubs, regional sequencing centers to do the sequencing and also even perform the bioinformatics analysis for timely reporting. So these are basically the genomics surveillance workflow that we are carrying out at the continent using the PGI 2.0. So what are the role of genomics in One Health? One Health is a big, interconnectedness between humans, animals, and environment. And as I mentioned in the previous slide, that there are lots of zoonotic diseases outbreak in Africa, particularly in western parts of the countries. And these zoonotic diseases, the key ones are Mpox, Ebola, avian influenza. They are actually, genomics are actually being applied to those pathogens. For example, like Mpox, genomics help to trace multiple introductions that is sustained between human-to-human transmissions, and also identify spillover from wildlife and track viral evolution in Ebola case. Avian influenza genomic surveillance actually reveals the clade expansions and interspecies jumps, how they are interconnected, between humans, animals, and even the environment itself. Genomics can also have a reach, that can help in health, in tracking the health pathogens, and also strategizing through the molecular health insight. So it helps us in understanding the real-time genomics data, enabling early detections and confirmations of novel and emerging pathogens in the countries. So genomics for diversity and surveillance, if you see the previous slide, what I mentioned was actually a loop that is being cycling in what we call anthropogenics. And it's also the same scenario that we apply here, identifying the species as environmental scientists within the ecosystems, like the biodiversity ecosystems. We monitor, the spread of the pathogens and what are the shifts, and then also detect some environmental DNA from the wastewater, soil, air, and wildlife. This is how you genomics can be applied, in monitoring or carrying out some surveillance systems in the entire biodiversity ecosystems through non-invasive biomarker discoveries, as well as biodiversity databases, linking what is sequenced at this country, what has gotten in the other countries. That will inform where the disease are actually coming from, and we can definitely track using the applications of genomics in these ecosystems. So back to our discussions on how it was started, the PGI, and where we found African countries or African Union member states in terms of functional NGS capacity. There were seven countries that were able to have, functional genomic sequencing capacity, as you see in the map by the left. That is the pre-COVID. After COVID, in 2024, we were able to, expand the foundational genomic sequence that we found in 2019, to this level. Now we have 41 countries that have full functional genomics capacity in all the national public health institutions, so it affiliates like research institutions. And as well, currently we are trying to, support the remaining 9 member states that are not having the capacity at the moment through, partnerships with SLMs and many other partners to catalyze these capacity expansions in the national public health institutions, as well as the national reference laboratories. But this is what we have as post-pandemics as of 2024. And moving forward, these countries, we are able to actually sequence, at least we had 39 countries that can, be able to sequence the SARS-CoV-2. 16 can have the capacity to sequence the SARS-CoV-2. At least one other virus is either HIV, influenza, or polio, measles. And also we have 6 countries out of 39 have the bacterial sequencing capacity, like E. coli, cholera, and the rest. And we also have 5 countries out of the 39 that have malaria, sequencing capacity in the continent. So public health resilience will actually help, or through genomics, public health resilience can be strengthened through 3 different areas, like early warning systems. Genomics can support the early detections of novel and reimagined pathogens within the ecosystems of the biodiversity, inform targeted interventions as early as possible before even the pandemics can go, track AMR, identify resistant genes, in the environment, monitor trends, across pathogens, and that really help in informing decision-making based on the obtained data at early warning systems, tracking AMR, that guides the vaccines and treatments, as well as the policy responses to the countries. So in terms of the data sharings, what Africa CDC recently developed a continental framework in each of the subdivisions that we have at Africa CDC. And that continental framework actually encourages member states to promote timely sharing of data and also coordinated sharing of data. And that aligns with the African Union vision for sovereign, trusted data governance. In terms of ethics, equity, and engagements, Africa CDC ensures community involvement and culturally appropriate consent is sought before the data is shared to the public, encourages benefit sharing with data contributors, especially from low resource settings in the countries, and encourage fair, findable, accessible, fair principles of data sharings and open science practices across the member state to support basically transparency, reproducibility, and collaborations. So the key challenges thus far we found is basically infrastructure limitations. There are still limited sequencing labs, cold chain, supply, and also internet as well as the power supply are the key challenges in the countries. And workforce shortages, scarcity in trained bioinformaticians, even though we frequently train bioinformaticians, but they frequently left the countries, to other advanced countries. Lack of scientists and as well as data analysts. Sustainability issues, because of our heavy reliance on the donor funding and vertical projects. So, but however, we have some opportunities that could possibly help those challenges, which I itemize here. The use of mobile genomics platforms like portable sequencers, MinION, enabled field diagnostics and surveillance, applications of AI in enhancing pathogen detections, early outbreak predictions, as well as analysis, multi-sectoral partnerships like strengthening the coordination across health, agriculture, and environmental sector in a way of, one health approach that could really help. So what are the future directions for this? I thought of this for cycles, to encourage strong linkages with biodiversity programs like Africa BioGenomes, Genome, Africa VGI, linkages with One Health applications, human health environment, broadening One Health applications as well as doing what we call integrated genomic surveillance network. So the first is basically to foster strong collaborations and linkages between biodiversity programs. Second, establish cross-border and multi-disease sequencing platforms across the national public health institutions, and also promote the co-development of bioinformatics tools and data sharing, develop SOPs and that could be helpful within the countries, and expand genomics beyond outbreak response, so like climate-sensitive diseases, food safety and agricultural pathogens, wildlife health and ecosystems integrity. So the basic recommendations, the way forward after looking at the challenges, to strengthen the inter-country collaborations. As we understand, most countries are not really, collaborating with one another, even though they are neighbors. So facilitate sharing of sequencing infrastructure and expertise will help in strengthening collaborations in inter-country and regionally, promote cross-border data exchange, outbreak coordinations, and regional workforce, regional networks, as well as harmonize genomics protocols. For example, countries may decide to, develop standardized SOPs and metadata frameworks across sectors, align surveillance workflow between humans, animals, and also environmental health, and support the workforce trainings and infrastructure for data analysis. And one other thing is very important is, I think, retention. We keep training scientists across the continent for genomics, bioinformatics, as well as sequencing, but they keep leaving the countries for a better future, because there are so many other challenges that are there within the countries. I think if the policymakers could, make use of their power, to find a way of retaining those bioinformaticians as well as the data scientists in the public health institution, that could really solve most of the problems and the challenges of data interpretations that we have in the country. So to conclude my talk, basically genomics is a cornerstone for One Health, particularly in Africa. So empowers early detection, surveillance, and response is the key. Links biodiversity monitoring to public health protections. So my call for actions is basically to foster cross-sectional, cross-sectoral and regional collaborations, inter-countries collaborations, support capacity building in genomics and bioinformatics, as well as align initiatives like Africa BioGenomes Project, Jumbo, Africa CDC, PGI, national strategies, because most of the African countries nowadays are being encouraged to develop their national genomic surveillance strategies. We have about 10 countries now that have so far developed their national genomic surveillance strategies, and that really align with the One Health approach that Africa CDC are aiming at. With that, I thank you for the opportunity to speak on behalf of the Africa CDC and the Center for Laboratory Diagnostics and Systems. Thank you so much, and over to you if there's questions. Dr.
Thank you.
Tambot.
Thank you, Dr. Umar Ahmed. I know there are lots of questions for you, or we are running late, so we're going to allow maximum 2-3 questions. If you want to ask your question to Dr. Umar Ahmed, please do raise your hand. And also, if you have any question for him, also please do feel free to put it in the chat box, and he will respond via the chat box if we can't accommodate it during this meeting. Olushola Thomas, could you please ask your question, please? Or Yaya or Josephine, anyone can unmute?
Yeah. Hello, everyone.
Can you hear me?
Okay. Yeah. You want to go ahead?
Yeah. Good afternoon, everyone. My name is Yahya Said. I'm a master's student from Anhui Polytechnic University, China. So my question is that how can we benefit, we that we are not in the Africa, but we are from West Africa, but we are studying abroad, not within the region of Africa. How can we benefit from the African CDC?
Dr. Amar?
Thank you so much, Yahya, for these questions. Actually, Africa CDC, operates based on, the mandate of the African Union, which means if you are from the Africa, it means you have potentials to benefit from the training and capacity building by Africa CDC. But it focuses more of supporting the national public health institutions. So if you are affiliated with any of the national public health institutions in Nigeria, let's say like Nigeria CDC, IState, you will definitely benefit with the ongoing fellowships in bioinformatics and genomics, ongoing trainings in sample repairs, and many of the number of trainings that we open organize. So you're Africans, you have, the potentials, you can, of course, benefit from all of those trainings whenever it's advertised. Sometimes we do send those requests through the National Public Health Institution directors to request for nominees. And if you are lucky to be nominated, then of course you